Video we are going to see about peanut mottle virus. The causative agent type is virus, family Partiviridae, genus Partyvirus, genome single stranded positive sense RNA virus. Morphology of the organism filamentous and flexuous rod shaped virus. Particle length is approximately 650 to 700 nanometers. Diameter is about 12 to 15 nanometers. Possess helical symmetry. Composed of a single coat protein of about 34 to 36 kilodaltons. Contains single stranded positive sense RNA genome. Genome size is about 8.4 to 8.7 kilobases. Similar morphology to other Kerala virus. Found in peanut plants. Observed under electron microscope in infected plant tissues. Host plant. Peanut mottle virus primarily infect leguminous plants. Its host range includes several important crops and wild species in the Fabaceae family. Primary host is Archaeopogea, main economic host and most severely affected. Other known hosts are soybean, common bean, cowpea, lima bean, velvet bean, Lesbodus species, Desmodium species, Wisteria species. Characteristics of peanut mottle virus disease belong to the genus Partyvirus in the family Partiviridae. Infects mainly peanut plants and some other legumes. Has flexuous filamentous particles. Virion length is approximately 720 to 750 nanometer. Contains single stranded positive sense RNA genome. Transmitted primarily by aphids in non-persistent manner. Also spread through infected seeds. Causes in host plants. Reduced pot yield and seed quality mottling. Chlorosis and stunted growth in peanuts. Detected using ELISA, RT-PCR or electron microscope. Virus replication and movement occur inside the plant cells, widely distributed in tropical and subtropical regions. History of peanut mottle virus disease. First identified in the United States in the early 1960s. Initially discovered in peanut crops showing mottling and leaf distortion. The virus was named after the distinctive mottle symptoms on peanut leaves. Early studies confirmed seed transmission as a major route of spread. Research expanded in the 1970s to 1980s include vector transmission by aphids, detected in other legume hosts including soybean and cowpea, spread to Africa, Asia and Latin America due to international seed trade. Diagnostic techniques such as ELISA and RT-PCR were developed in 90s. Still poses threat to peanut production in tropical and subtropical regions. Control efforts focus on seed certification, resistant varieties, and vector control. Transmission. Transmission was done by AP transmission, seed transmission, mechanical transmission, and host to host contact. In AP transmission, spread by aphids like Apis carcinoma in a non persistent manner. Seed transmission. Virus can be passed through infected seeds, especially in peanuts and soybeans. Mechanical transmission. Mechanical transmission was done by during handling, cultivation, or contaminated tools. Host to host contact is through close contact between infected and healthy plants. Pathogenesis of peanut mottle virus. Virus enters the plant through aphid feeding or infected seeds. Infects epidermal and mesophyll cells at the site of entry. Viral RNA is uncoated and translated to produce replication proteins. Replication occurs in the cytoplasm using host cell machinery. Produce a replicase complex that synthesizes new viral RNA. Forms inclusion bodies with infected cells. Viral RNA and proteins move from cell to cell via plasmodesmata. Long distance movements occurs through the phylum tissue. Virus disrupts normal cell function, causing chlorosis, mottling, and reduced growth. Accumulation of virus leads to systemic infection in the whole plant. Symptoms. The symptoms of the virus in peanut appears in irregular dark slants and young leaves. The symptoms are not as clear and older leaves and thus can be easily missed even when the virus is epidemic proportion in the field. In infected plant cells, the virus makes characteristic spotty virus cylindrical inclusion that are visible in the light microscope with proper staining. Treatment: no direct chemical treatment is available to cure infected plants. Use of virus free certified seeds to prevent seed-borne infection. Grow resistant to tolerant peanut varieties if available. Control aphid vectors using insecticides or biological methods. Practice crop rotation with non host crops. Avoid planting near infected legume crops to limit cross infection. Maintain field hygiene and sanitation to reduce virus sources. Control: use healthy and virus free seeds. Grow resistant peanut varieties. Control aphids using insecticides. Rotate crops to avoid continuous peanut planting. Remove and destroy infected plants. Clean tools and equipments regularly. Remove nearby legumes, weeds, and wild host. Preventive measures: use certified disease free seeds for cultivation. Avoid planting peanuts near infected fields. Control aphid population early. Grow resistant peanut varieties. Remove weeds and wild legume plants. Clean tools and hands after handling plants. Avoid planting the same crop repeatedly in one area. Thank you.